The biggest eureka moment was the realization that by developing molecules to inhibit DNA repair, you could selectively kill certain cancer cells and not affect normal cells. And that was a real realization that was a huge moment for me, and actually then set the path for the development of olaparib. Olaparib is what we call a small molecule drug. It's a chemical compound that can be formulated, for example, in pills, and then can be taken by cancer patients. And the remarkable thing about olaparib is once it goes into cells of the body, it inhibits a key process of DNA repair by engaging with a protein called PARP, P-A-R-P. Now that doesn't have an effect on most normal cells because they have resilience and they can get around that inhibition. But what this remarkable drug does is that it will kill cancer cells that have defects in another pathway driven by proteins called BRCA1 and BRCA2. It works by killing the cancer cells but leaving the normal cells of the patient unscathed. So I worked in the Gurdon Institute for many, many years. It's where I first started out as a group leader way back in the early 1990s. My lab actually stumbled into the field of DNA repair. It wasn't something we were actually looking to do, but we had some interesting observations that took my lab into that domain. And we've been working on DNA repair and related processes ever since. So in the early days in the Gurdon Institute, we realized that some of these DNA repair factors that we're working on were potential drug targets. And that led me to conceive of the idea of developing drugs to inhibit DNA repair. The next big eureka moment, if you like, was finding that those compounds were selectively killing certain cancer cells, but not affecting normal cells in tissue culture. And this really galvanized me and the team in our research lab, but also in the company that I'd founded. Kudos to translate those molecules into the clinic and see whether or not they would affect patients in the right kind of way. We were time and again excited and astounded by the fact that this idea was actually being borne out. So the company that we set up in a relatively short period of time managed to develop this compound, olaparib. We managed to formulate it, and then we actually took it into the first human individuals without any toxicity. And this was a remarkable achievement in its own right. But of course, then we're in a situation where we knew that we needed to explore this molecule in clinical trials, and they clearly involve lots and lots and lots of work, patients, and associated costs. And we realized that we needed to raise substantial sums of money in order to do that. And so at that stage, we realized that we needed to talk to pharmaceutical companies to see if we could partner with them. And one of those companies were very interested in the program, but actually turned around and said, well, actually, we'd like to buy the whole company and then take forward this program as well as others within. That company was AstraZeneca, and they took olaparib forward. That compound is now with a brand name Lynparza, and that's been brought through clinical trials by AstraZeneca in partnership with Merck. And it's amazing that over the course of the last few years, this drug, which started out in a small biotech company, is now being used to treat many, many thousands of patients across the globe.
So olaparib is really the first PARP inhibitor that came into the clinical community for wider usage. For patients who have hereditary changes in the BRCA gene, it's incredibly important. It's such a valuable additional tool. I've used olaparib in a clinical trial called PARTNER, and PARTNER is a trial for early stage breast cancer patients with either triple negative or BRCA positive breast cancer. And olaparib has been used in combination with chemotherapy in those patients, and for the BRCA patients, it's been tremendously successful.
Our research focuses on trying to personalise treatment for women with ovarian cancer. And that's a major challenge because the wiring diagram, in other words, the DNA and the makeup of cancer cells in women with ovarian cancer, is very complex. So we're using methods that use whole genome sequencing and more sophisticated methods to try and really choose the right treatment for a patient. So part of my job is also to work in the clinic looking after individuals with ovarian cancer, and we use PARP inhibitors, of which olaparib is one, as routine care now for patients. If we look at the results for individuals who've developed ovarian cancer because of a faulty gene, BRCA1 or BRCA2, and in those patients who take olaparib, they have a 70% reduction in the risk of dying or their cancer returning. So that means 70 ladies out of 100 are getting huge benefit. And this has been a dramatic advance in helping to prevent ovarian cancer recurring and helping patients stay free of symptoms.
My name is Melanie Newman. I was diagnosed in November 2018 with stage 3 high-grade serous ovarian cancer. I know that I'm really fortunate to be able to go onto olaparib and to tolerate olaparib so well. And it's made an amazing difference to my life because life is normal again now, and I'm assured and confident that olaparib is keeping the cancer away. Having had cancer, I do look at life differently now, and I want to do as much as I can with friends and family and just enjoy every moment.
I love being a scientist. I've always been a biologist, a scientist at heart, and I consider myself to be very fortunate that I can come to work in the morning and actually do something that I enjoy. The thing that excites me most is that we now have at our fingertips a set of techniques, approaches, computational tools that I wasn't even dreaming about 10 years ago. And so that gives us opportunities to do new types of science and take our research into new directions. And in addition to, I think, this new science telling us about DNA repair and the fascinating aspects of cell biology it controls, but I think this is going to point to new opportunities to translate that knowledge towards patient benefit.